Hello and welcome back to the Biochemistry for Health Sciences channel. In today's video, we shall discuss the extrinsic pathway of apoptosis using TNFR1 as an example. In the last video, we saw how the intrinsic pathway leads to the activation of executioner caspases, and in a future video, we'll see how executioner caspases go on to cause apoptosis or cell death. In this video, we shall look at how extrinsic apoptosis will cause the activation of executioner caspases, and then that executional caspases will cause cell death by apoptosis. Let's first look at examples of signals or ligands, as well as the receptors that are involved in extrinsic apoptosis. One example of receptor and their respective ligand involved in extrinsic apoptosis is the tumor necrosis factor family. So this includes, for example, the receptor tumor necrosis factor receptor 1 or TNFR1, which binds the ligand TNF-alpha. Then we have the FAS receptor that binds the ligand FAS ligand. And then we have the TNF-related apoptosis inducing ligand receptor, such as TRAIL-R1 and TRAIL-R2. And these bind the ligand TRAIL. We have then the dead receptors like DR3 and DR6 that bind ligands like TNF, like cytokine A, and amyloid precursor protein or APP. Then we have other receptors that are involved in apoptosis. Toll-like receptor 3 that binds double-stranded RNA. Toll-like receptor 4 or TLR4 that binds lipopolysaccharide, bacterial lipopolysaccharide. And then we have the zDNA binding protein 1 or ZBP1 that binds the Z form of nucleic acids that are produced by some viruses. In this video, we shall use this TNFR1 and its ligand TNF-alpha to show how this receptor ligand combination leads to extrinsic apoptosis. In the next video, we shall look at the FAS receptor, which is very similar to the TRAIL receptors, and their respective ligands, FAS ligand and TRAIL ligand. And in a future video, we shall look at the other receptors, such as the toll-like receptor, as well as the zDNA binding protein 1, which trigger extrinsic apoptosis. So here today we will look at this. The TNFR1 is the receptor and the ligand is TNF-alpha, and we'll see how this leads to the activation of the executioner caspases 367, and that eventually leads to apoptosis. As mentioned in an earlier video, cell death is a very complex process. Involves many different proteins. So we'll try to keep this as simple as possible and try to look more at the concepts, the big picture, and we'll only discuss proteins that we know of very well today and that are medically important. Now, the TNFR1 pathway can trigger the cell to survive. So it can trigger pro-survival, pro-inflammatory program. So the cell survives and there's inflammation, or it can cause cell death. Instead of the cell surviving, it can kill the cell. And the cell can die either by the extrinsic pathway of apoptosis, or there is another program where the cell can die by necroptosis. Okay, so apoptosis doesn't have much inflammation. Necroptosis is associated with inflammation. So this TNFR1 pathway can cause three very important programs to be activated, either survival or cell death by apoptosis or by necroptosis. So let's first discuss the survival program. Okay, so this is the program. We'll talk about this first, and then we'll look at how the cell can go through this pathway or even through necroptosis. The actual necroptosis pathway we'll talk about in a future video. Before we discuss the pathway, let's look at some general points about the ligand tumor necrosis factor and their receptors, the TNF receptors. So these ligands, the TNF, belong to what we call the TNF superfamily, sometimes abbreviated as TNF-SF. And there are about 19 ligand cytokine-like molecules or ligands that we know of today. These ligands are expressed mainly by cells such as macrophages, natural killer cells, the T and B lymphocytes, basophils, eosinophils, endothelial cells, smooth muscles, and a few other types of cells. Now, this name is kind of interesting. Tumor is a tumor, sort of a growth or cancer, and necrosis is killing the cancer cell. Factor is just a chemical, or in most cases protein. So a protein that kills cancer is kind of an interesting name. And this name came, if you look at the history of this, in around 1891, the physician William Coley, he observed patients who had certain bone tumors. These bone tumors actually disappeared, they regressed after they had certain infections. For example, he observed patients with a skin infection had their bone tumors regressed. So that was kind of a interesting observation. And in 1975, this factor was identified and was called as tumor necrosis factor. The actual thing that caused the tumor necrosis factor to be released was the lipopolysaccharide from the bacteria, from the infection. So TNF, these are very powerful, very potent pro-inflammatory molecules. So there are 19 TNF ligands, but the receptors, the TNFR superfamily receptors, there are about 29 such receptors that we know of that bind the different types of TNFs. And these receptors are expressed by various cells, such as the hematopoietic cells, immune cells, endothelial cells, epithelial cells, hepatocyte, lung, colon cells, natural killer cells, and so on. Many, many cells express the TNFRs. Now, several ligands can bind to more than one receptor, and some receptors can bind more than one ligand. This helps form a very large communication network, and this helps to regulate the immune system as well as homeostasis. So this whole TNF receptor family is a very important family of proteins that regulate the immune system as well as several things in homeostasis. For example, the TNF-6 signaling is involved in cell survival, in cell proliferation, in differentiation, and of course in the immune system, one of which is the important function of the immune system is the defense against pathogens such as bacteria, viruses, parasites, and so on. Well, of course, scientists were very excited that William Coley observed that cancers can regress after an infection, and TNF was the chemical responsible for it. So, of course, logically, what scientists did, they treated animals with TNF. However, there was a serious problem. TNF is a very powerful inflammatory chemical, and injection of TNF can lead to a very deadly inflammatory shock syndrome. So therefore, TNF really cannot be used for cancer treatment. However, TNF inhibitors are being used to treat chronic inflammatory diseases and inflammation due to autoimmune diseases such as rheumatoid arthritis and inflammatory bowel disease, because TNF is such a powerful inflammatory molecule. So inhibitors of TNF can be used to suppress inflammation. And since inflammation is a very important topic in pathology, is responsible for so many diseases, clearly this TNF family of ligands and receptors is a very, very important process to talk about, and that's why we will try to mention as many proteins as possible, not to confuse you, but to try to appreciate that these proteins you might come across in your future as a healthcare professional, because they are related to many, many human diseases. So since there are so many different TNF ligands and receptors, in this video to explain the concept, we will focus only on the one example, and where the ligand is TNF-alpha and the receptor is TNFR1. The receptor TNFR1 is found predominantly in hematopoietic cells, immune cells, as well as endothelial cells. And TNFR1 binds the signal TNF-alpha as well as a very similar molecule called lymphotoxin alpha or LT-alpha. The ligand TNF-alpha is a cytokine that is expressed by cells such as macrophages, natural killer cells, and T and B cells, while lymphotoxin alpha is expressed mostly by NK, T, and B cells. So from this you can see that this combination plays a very important role in the immune system. Okay, so let's look at this signaling pathway that leads to first cell survival. So in the first step, as always, the ligand will bind the receptor. Now the receptor TNFR1 has a cysteine-rich domain, a CRD, and this domain will bind the ligand's TNF homology domain, or THD. So that interaction will cause the ligand to be bound to the receptor. Besides the cysteine-rich domains, TNFR1 also has the transmembrane or TM domain that allows the receptor to be embedded in the plasma membrane, and it also has a DET domain. So when TNF-alpha ligand binds to the receptor changes shape and its DET domain gets exposed. And once this DET domain of TNFR1, once the DET domain is exposed, now it will initiate the formation of what we call complex 1. Now, the dead domain of the receptor will now bind the dead domain of a protein called THREAD, which stands for TNFR1 associated dead domain. So you can see THREAD has a dead domain also. So this interaction of dead domains will recruit THREAD to the receptor. So now we have THREAD that is bound to the receptor. Now THREAD is an adapter protein for the protein RIPK1, a very important protein to remember, RIPK1, which stands for receptor interacting protein kinase 1. Now RIPK1 has a kinase domain. Remember, kinase is an enzyme. So it has an enzymatic activity, a KD domain. You can see the KD domain here. Then it has an intermediate domain that contains a RIM domain, which stands for RIP homotypic interaction motif. So then you see the RIM domain, and RIPK1 also has a dead domain, DD. Okay, so we see the enzyme domain, the RIM domain, and the dead domain. And recall from a previous video, it's important to know these domains because these proteins use the domains to help bind to other proteins. So now the dead domain of THREAD will interact with the dead domain of RIPK1, and RIPK1 will now be recruited to complex 1. Now THREAD also has the N-terminal domain, and this N-terminal domain of THREAD will interact with the C-terminal domain of another protein called TREF2, and this protein stands for TNF receptor associated factor 2. So this interaction will lead to the recruitment of TREF2 to complex 1. So now TREF2 will be bound to THREAD, and you can see slowly the complex 1 is being formed. Now TREF2 is an adapter protein for CIAP1-2. So now CIAP1-2 will join complex 1, and this will now complete the formation of complex 1. Now CIAPs are members of inhibitors of apoptosis, the IAP family that we talked about in a previous video. Now the CIAPs have a domain called a ring domain, and this ring domain confers to this protein's ubiquitin ligase activity. So since CIAP can add ubiquitin chains, that's what it does. It adds K63, lysine 63 ubiquitin chains to RIPK1. So this RIPK1 now has these ubiquitin chains. This is a very important step for the cell survival program. Ubiquitin chains are also added to other proteins such as THREAD. We talked about this ubiquitination in video 5.28. So for a refresher, you may want to look at video 5.28 for this ubiquitination process. Now, unlike K48 ubiquitin chains added to proteins, K63 linked proteins do not undergo proteasomal degradation. Instead, they will help the RIPK1 to recruit other proteins. So by adding this ubiquitin chain to RIPK1, it's not destroyed. It actually allows it to recruit other proteins. So now RIPK1, that is ubiquinated, will recruit a multimeric protein complex known as LUBEC, linear ubiquitin chain assembly complex, and it will also help recruit a ubiquitin binding protein called TEC1, which stands for TGF-beta activated kinase 1. So now we have Lübeck recruited by RIPK1 as well as TEC1. Now Lübeck adds M1-linked linear polyubiquitin chains to various proteins such as TNFR1, TRED, as well as RIPK1. And this addition of M1-linked chains stabilizes complex 1. So now we have a very stable complex 1. Just as a side note, M1-linked polyubiquitin chain has ubiquitin molecules linked as a linear chain via peptide bonds, not isopeptide bonds as we talked about in an early video, as you see in K48 or K63. So here we're talking about peptide bonds. So it's a different type of ubiquitin chain, a linear format. So in complex 1, we have this very complicated ubiquitin chain network. You can see a very complicated network of ubiquitin chain. And this ubiquitin chains, they act as a scaffolding, and that allows for recruitment of other proteins. And one of the proteins that is recruited is this IKK complex. Okay, that stands for inhibitor of kappa B or IKB kinase, so IKK complex. And this IKK complex contains IKK alpha, IKK beta, as well as a ubiquitin binding subunit that is called NEMO. NEMO stands for NF kappa beta essential modifier, also known as IKK gamma. So take-home message here is now this ubiquitin chain allows IKK to attach to this complex. What happens in the next step? The TAK1 kinase activity of this protein will now phosphorylate IKK beta present in the IKK complex, and this phosphorylation is going to activate the IKK complex. The activated IKK complex will then stimulate the translocation or the movement of NF kappa B to the nucleus. And once it goes to the nucleus, it will induce NF kappa B dependent transcription of various genes. Meanwhile, TEC1 will trigger the mitogen activated protein kinase pathway or the MAP-KK pathway. So now we have NF-KB transcription, mediated transcription, as well as the MAP-KK pathway that is stimulated. The outcome of this NF-KB transcription as well as MAP-KK pathway is the increased expression of pro-inflammatory genes such as interleukin genes, TNF genes, chemokine genes, and all of this leads to proteins that will cause inflammation. And there's also increased expression of genes for survival, pro-survival genes such as the anti-apoptotic protein genes, as well as the CFAR gene, which encodes the protein C-flip that can also act as an anti-apoptotic protein under certain circumstances. And we'll talk about C-flip later on. So the end result of this is pro-inflammatory, pro-survival program. So the cell survives and there is inflammation. In order for the cell survival program to occur, it's important that the kinase activity of RIPK1 is inhibited. So the kinase activity of this enzyme RIPK1, if it's inhibited, then the cell will survive. And this inhibition is done by phosphorylating RIPK1. And this phosphorylation is carried out by several different means, by the proteins TBK1 and IKK epsilon, as well as the IKK complex and the MK2. It's also known that the K63 ubiquitination of RIPK1 helps in its phosphorylation and therefore inhibition of its kinase activity. So if any of these activities does not happen, is suppressed, then TNFR1 signaling will lead to cell death instead of cell survival. Okay, so that's how TNFR1 signaling leads to the pro-survival, pro-inflammatory program. Now let's look at this part here. How does TNFR1 cause apoptosis by the extrinsic pathway? And if that doesn't happen, then it can cause necropolis. So as mentioned earlier, RIPK1 plays a very important role. Whether the cell survives or dies seem to depend on several factors, including the ubiquitination status of RIPK1, as well as the caspase activity. Now, if RIPK1 is ubiquitinated, then that makes complex 1 very stable and the cell will survive. However, if RIPK1 is not ubiquitinated, or if the ubiquitin chains from RIPK1 are removed, then complex 1 is unstable, and now the non-ubiquitinated RIPK1 can dissociate from complex 1, which is in the membrane. Remember, all this is part of the plasma membrane. And now this RIPK1 can leave the plasma membrane and enter the cytoplasm and start forming what we call complex 2, a death-inducing complex 2 in the cytoplasm. So this modification of this ubiquitin network is associated with TNFR1 is important, and how this is actually done seems to be very complex, and until now we don't completely understand this process. One of the proteins that seem to help RIPK1 dissociate from complex 1 and enter into the cytoplasm to form the death-inducing complex 2 is a deubiquitinating enzyme called CYLD. CYLD stands for cylindromatosis. So this is a deubiquitinating enzyme. CYLD removes the M1K63 ubiquitin chains from RIPK1. And this CYLD is recruited to this complex by another protein called SPETA2. SPETA2 stands for spermatogenesis associated 2, which binds the protein Lübeck. So SPETA2 joins Lübeck, and then SPETA2 brings in CYLD, and the CYLD now removes the ubiquitin chains from RIPK1, and now the RIPK1 can now leave the unstable complex 1 and leave the membrane and enter the cytoplasm to form complex 2, what we call the death-inducing complex 2. As a side note, there are some proteins that can inhibit the CYLD activity. So, for example, A20 which is a TNF-alpha-induced protein 3, as well as ABIN. So A20 and ABIN can actually inhibit CYLD activity. So these proteins prevent RIPK1 from entering complex 2. So these proteins actually promote cell survival rather than cell death. So once RIPK1 is able to free itself from complex 1, the unstable complex 1, and now this RIPK1 enters the cytoplasm, and once it's in the cytoplasm, it will recruit this protein TRED, TRADD. Now TRED has the dead domain, RIPK1 also has the dead domain, and so this RIPK1 TRED will now induce the recruitment of another protein called FADD, which stands for Fas-associated dead domain, because FADD also has the DD domain. So through these DD domain interactions, FADD is recruited to the complex 2, the death-inducing complex. Now FAD is an adapter protein for caspase 8 as well as the protein CFLIP. We'll talk about this CFLIP in a few minutes. So now we have the DD of FAD. You can see the DD of FAD interacting with the DD of procaspase 8 as well as the DD domain of this protein CFLIP. And so through this interaction, CFLIP and procaspase 8 will now attach to FAD, which is part of complex 2. So this attachment of CFLIP and caspase 8 to complex 2 now completes the death-inducing signaling complex. So now we have this DISC formed in the cytoplasm. As a side note, overexpression as well as decreased expression of this protein FADD has been observed in several types of cancer. Now, caspase 8 has two DD domains. So the DD of FAD binds one of the DD on the procaspase 8, and that will expose the second DD on procaspase 8. And this second DD of procaspase 8 will allow it to bind to another caspase molecule, which will then expose the other DD of the new caspase molecule. And this chain reaction will then lead to the formation of DD oligomers or filaments composed of caspase 8 as well as the protein CFLIP. Now, in this case, CFLIP-L, we'll talk about it in a few seconds, the long isoform, acts as a pro-apoptotic protein. Okay, we'll see that CFLIP can act as either pro-apoptotic or anti-apoptotic. In this case, it acts as a pro-apoptotic protein because it stabilizes the active site of caspase 8. So we have this active caspase 8 now, and this active caspase 8 will now go on to cleave and activate executional caspases 3-7. Okay, so now the extrinsic apoptosis pathway will cause the activation of executional caspases 3-7, which will then go on to carry out apoptosis. Now, caspase 8 also destroys RIPK1 activity. Okay, destroys the RIPK1, cleaves RIPK1, and this also helps promote apoptosis instead of necroptosis. We will see in a few seconds, RIPK1 is also very, very important for necroptosis. But by getting rid of RIPK1, we are promoting apoptosis and not necroptosis. Now a few words about this protein C-flip, which stands for cytoplasmic Flies inhibitory protein. Flies is the abbreviation for fat-like IL-1 beta converting enzyme, which is one of the many names for caspase 8. Now the C-flip protein has 3 isoforms. We have the long isoform, about 55,000 Daltons molecular weight. This is called C-flip with an L. It has 2 DD domains, and then of course a pseudocaspase, so it looks like a caspase but it doesn't have any activity, domain, pseudocaspase domain. And then there's the short isoform, about 27,000 Daltons molecular weight. So this is the short isoform. It has the 2 DD domains, but it doesn't have the caspase domain. And then there's a regulator isoform, about 27,000 molecular weight, with the R here. This too has 2 DD domains without the pseudocaspase domain. So there are 3 forms of C-flip. Okay, the C-flip L, C-flip S, and C-flip R. Note that only this form has the pseudocaspase domain, but it has 2 DD domains. This also has 2 DD domains, and this also has 2 DD domains. Now it seems like C-flip can act as an anti-apoptotic protein that promotes cell survival, or it can act as a pro-apoptotic protein. So in the previous example, we saw how C-flip at the death-inducing complex acts in a pro-apoptotic way by formation of procaspase 8 C-flip heterodimers, where the C-flip stabilizes the active site of caspase 8, thereby promoting apoptosis. So in this example we saw in just a few seconds ago, C-flip L acts in a pro-apoptotic way. Now C-flip, any of the forms, all 3 forms, because all 3 forms have the DD domains, so all 3 forms can also competitively inhibit binding of caspase, procaspase 8 to FAD. So caspase 8 binding to FAD is done through the DD domain, but because these forms also have DD domains, so if we have high amounts of these forms, they will compete with caspase 8 for binding to FAD. And thereby, in this case, they can prevent the activation of caspase 8 by limiting the growth of the DD filaments, thereby inhibiting apoptosis. Okay, and in this case, actually promoting cell survival. So C-flip can promote cell survival or promote apoptosis. Okay, so far we have seen how TNFR can lead to cell survival, and we just saw how TNFR can lead to extrinsic pathway of apoptosis. So what about necroptosis? How is this pathway triggered? So here we have the cell survival program. Here we have the apoptosis. What about necroptosis? Well, necroptosis happens, the cell can undergo necroptosis if caspase 8 is inhibited. Okay, if we inhibit caspase 8, for example, by flip. So you can see viruses that produce a lot of flip, they can stop the cell from undergoing apoptosis. Okay, so caspase 8 is inhibited, RIPK1 is not destroyed. Okay, so if RIPK1 is not destroyed, if caspase 8 is inhibited, Now RIPK1 will recruit RIPK3, that will recruit this protein MLKL, that will now trigger necroptosis. Okay, so viruses and cancer cells that inhibit apoptosis can still undergo necroptosis. So this is a very nice program to help cells that are infected with viruses or cancer cells and help them actually die to protect the human. So once again, since many viruses produce inhibitors of caspase 8, necroptosis probably evolved as an antiviral defense mechanism. Make sure that these cells that are infected with viruses and are stopping this apoptosis, the cell has an alternate program, and that is necroptosis, to die. Okay, we'll talk about necroptosis in a future video. So to summarize, this is how this very complex but important TNFR1 signaling causes either the cell to survive with inflammation, or causes the cell to die by the extrinsic pathway, or leads to cell death by necroptosis. Important difference is this doesn't have inflammation. This you have inflammation. Necroptosis, you have inflammation. In the next video, we'll look at another TNF family just to explain this very important concept. And we'll use the example of the FASR receptor, which is very similar to what we will see with the TRAIL-R receptor. Okay, so until the next video, once again, thank you very much for watching. Goodbye, have a wonderful day. Hopefully we'll see you soon.